Hello, in this video we will discuss about the Peyronie's disease. So what is the Peyronie's disease? That is the reproductive disease which that will lead to damage the penis of the male reproductive organ. So the Peyronie's disease is short for the PD, is a benign tumor formation and progressive fibrotic disorder characterized by scar or plaques within the tunica albuginea of the penis. So benign tumor formation means it will not spread throughout the body like a malignant tumor. So here is the reproductive organ which that is show the corpus cavernosum filled with blood during the erection time, and it is a fibrous plaque will be developed in the upper part of the penis which that contain corpus cavernosum filled with blood. So it will be bent upward during the erection time. So let's cross section of the penis is looking like this and contain a corpus cavernosum, and the tunica albuginea is the covering with the corpus cavernosum, and inside the corpus cavernosum is the trabeculae and cavimosal spaces. So here is the urethra, which that is a corpus spongiasum is surrounded by the urethra. So that's all muscles is basically is used for the erection without bones. So here is the genetic factor responsible for fibrogenesis. So that's fibrogenesis formation, that fibrous tissue formation due to the growth factor, and semi-erectile dysfunction, as well as the possibility of the masturbation. So let's begin to understand. Here is the structure which that is the damaged penis due to the Peyronie's disease, which that is the firm outer casing where plaque forms, and Peyronie's disease formation, and in this way the here is the corpus, and the corpus cavernosum filled with blood will be bent. So that is why here the Peyronie's disease which that is, and here is a normal healthy penis, we can say that is the erect and not bended. So let's begin to understand the pathological stimuli and mechanical stress. During the intercourse, that is mechanical stress due to the semi erection of the penis, which that will lead to damage the upper part of the, and the growth factor which that will lead to proliferation of the cell, and the fibrogenesis will be occur, and the chronic inflammation, reactive oxygen species and reactive nitrogen species, cytokines, which that is responsible for this fibrogenesis, and the chromosomal abnormality also. So the pathological factors is due to the age 40 to 70, 0.4 to 20.3% is increased by the age 40 to 70. So on the other hand, the diabetes mellitus 2 and erectile dysfunction, and this all thing is basically that transforming growth factor 1 beta 1 is a crucial cytokines in pathogenesis, and high level of the TGF beta expression cause the differentiation of fibroblast into a myofibroblast. So this fibroblast convert into the myofibroblast, and this is the excessive accumulation of extracellular matrix, especially collagen 1, 3, 4 types in penile plaques will be developed, and the plaque will hard to bend upward or side. So all these things we will discuss about with the cellular mechanism and how the cell will proliferate and differentiate into the scar tissue formation, and that will be after that it will be died through apoptosis due to the non-functional cells, and that apoptosis will lead to bend throughout the life. So this is the plasma membrane. This plasma membrane contains different receptor. This all receptor is used for this Peyronie's disease, but here is I am drawing just four receptor for understanding, because it is used for the understanding the overview. So this is the transforming growth factor beta 1 receptor, which that will be receive the transforming growth factor beta, and this is the fast receptor which that is used for the apoptosis. So this is the death receptor, and the death ligand, the false ligand, will be bound with it for the apoptosis, but it will be inhibit. How it's possible? Let's begin to understand. First of all, we should need to understand the transforming growth factor beta 1, which that will be bound with the tetrameric protein, intrinsic protein, and the intracellular domain and extracellular domain. Intracellular domain is used for the phosphorylation, while the extra is used for the ligand binding. So is the phosphorylation will be occur, and the cascade of phosphorylation events will be occur, and this inactive SMAD P SMAD2 and SMAD3, which that is combined to dimerize, and this dimerization is known as the SMAD2 and SMAD3 complex. And in this way, this is also known as the SMAD pathway. So the non-SMAD pathway is basically P38 activation protein and Janus kinase activation, a RAS protein activation, and ARCS PI3K pathway, and AKT and GTPases, Rho A and CDC42. This all things is used for the tumor transforming growth factor beta induced cell response. So here the Rho stimulate ROCK, and ROCK will inhibit the ENOS, which that is the no production. So on the other hand, here you can see this is the transforming growth factor beta induced cell response. After this, the cell response basically is the activation, and that we will discuss later. But first of all, we should need to understand another pathway. So this is the RAS, which that it will be bound with the cytoplasmic membrane, and after this is inactive the GDP, and activated by this phosphorylation event to GTP. GTP used to basically the production, and in this way the activation of the RAS, which that will be stimulate the PI3K pathway and the RAF pathway, the activation of both protein. This will lead to RAF, basically it's mitogen activated kinase protein, and the activated AKT protein activation, and in this way both activation is used for the proliferation of cell. By the ARC1 and 2 activation will bind with the DNA, and in this way the transcription factor will lead to proliferation of the cell as well as the differentiation, and there are many things and enhance the proliferation. So how it's possible? Here is the SMAD pathway, which that is the phosphorylase, and SMAD4 will also bind to become a transcription factor. This complex will bind with a specific region of the gene for the proliferatory gene or growth gene, and in this way here is the transcription factor will bind with a co-activator. After the co-activator binding to transcribe this gene into the RNA, and this will lead to increase the transforming growth factor beta target genes. There are many targets, so the induced cell response. So in this way here is you can see this is also activate the BCL2 genes. BCL2 gene is very important and responsible for the and there are epigenetic mutation is also involved for the Peyronie's disease. So here is ARC1 and 2 is also responsible for the production of the BCL2 genes. This BCL2 gene is basically growth, cell cycle, enhance the proliferation, and therefore the rapid proliferation will be occur, which that is the benign tumor formation rather than the malignant tumor. So here is the AKT is also inhibit the glycogen synthase kinase 3 beta and MDM2, and this MDM2 stimulate basically, and mTOR pathway stimulate the mammalian target of rapamycin, and in this way here you can see this will be inhibit the growth, and the, basically the its growth BCL2 is also inhibit, and in this way the inhibition of the P53 protein, which that is used for the apoptotic gene, and the apoptosis will be inhibited by the inhibition of the P53 gene, while the activation of the mTOR gene to produce the mTOR complex. Here another is the interleukin 6. Interleukin 6 is the cytokines which that is bind and the recruitment of the JAK and STAT3 protein. By the docking of the JAK docking with the STAT3 phosphorylation will stimulate again the phosphoinositide 3 kinase, and in this way here is the extracellular and intracellular matrix. So the IECM and ICM, which that will be here is the stimulate the STAT3 will stimulate the PI3K, again AKT stimulation, and here the dimerization of the STAT3, and after the dimerization, after the phosphorylation will bind as a transcription factor, bind with a specific region of the gene for the stimulation of the mTOR pathway further for the proliferation of the gene, proliferation of the cell, and the maturation of the cell, and the differentiation of the cell. On the other hand, here is the growth factor. There are many growth factors. For example, insulin like growth factor or insulin, or there are many which that is bind to growth retinoblastoma protein uh, 2, which that is basically bind and the recruitment of the RAS and so son of 7 less, which that will be activated to inhibit the FOS ligand. FOS receptor is used for the death, and apoptosis will be inhibited by this mechanism and the transforming growth factor beta 1 receptor inhibition due to the activation of their receptor. So the FOS ligand will not bind. Here is the mTOR pathway. mTOR, the p53 protein will be inhibited while the mTOR pathway is used for the fibrosis, cell collagen increase, and extracellular matrix will be increased. That's all will be lead to Peyronie's disease. The fibrosis will be occur, and after this fibrosis is basically clear, after the apoptosis and the immune cell activation will lead to clear, but the damaged area will bend remain constant. So thanks for watching. Please make sure to subscribe, like, and share. Bye.